Hello and welcome to your monthly update from the COVIDENCE UK study. My name's Adrian Martineau and I'm the Chief Investigator based here at Queen Mary, University of London. So in today's webinar, I want to share with you some recently published data from another research group looking at the question of whether long COVID is associated with persistence of the SARS-CoV-2 virus. So let me explain a little bit of the background here. Now, although the presentations of long COVID are numerous, with some people predominantly suffering fatigue, others breathlessness, and others a whole range of symptoms in different body systems, essentially the major underlying pathology, or what's going on that's wrong in the body, seems to be a dysregulation or dysfunction of the immune system. This is in very general terms, but that dysfunction could arise really in two particular ways, and there's controversy about how that happens. The first hypothesis or idea is that after a SARS-CoV-2 infection, in people who go on to develop long COVID, their immune system reacts to the virus and continues to react in a dysfunctional way, despite the virus having been cleared from the body. But the alternative or competing explanation is that in the same way we're seeing immune dysregulation, but actually that is due to the SARS-CoV-2 coronavirus persisting in the body. So we have these two competing explanations, the one saying that the virus has been cleared, the other saying that the virus is persisting and is driving immune dysregulation. And what I want to present to you today is a findings of a recent study that suggests that perhaps that second explanation could be the right one. So the study is published in a journal called the Journal of Medical Virology, just published this year. The first author is Vaughan Craddock, and the investigators come from the University of Kansas Medical Center in Kansas City, USA. And the design of the study is as follows. The investigators recruited 47 people who had developed swab test positive SARS-CoV-2 infection. So that was proven either with RT-PCR or with a lateral flow test. And they followed up these people after their infection for a number of months. And 14 of them did not go on to develop long COVID, and 33 of them did go on to develop long COVID. The investigators then tested the serum, which is the non-cellular component of the blood, for the presence of RNA, that's genetic material from SARS-CoV-2 virus, and spike protein. So let me just remind you what a coronavirus looks like. And what you can see here highlighted in red are the two targets that were investigated in this study. One is the spike protein, which is that globular protein on the outside of the virus, and the other is RNA, which is the genetic material of the virus, which sits inside the virus. So the spike protein can be detected using a test such as the lateral flow test, or an ELISA, which stands for an enzyme-linked immunosorbent assay. And the RNA can be detected using a test called reverse transcriptase PCR, or RT-PCR. Often people refer to PCR, but actually for RNA, it's RT-PCR to be perfectly correct. So what were the results? Well, first of all, let's look at the outcome of the viral genetic material, the RNA that was detected at follow-up. In the patients who went on to develop long COVID, 54.5% of them still had viral genetic material detectable in their blood at follow-up, compared with just 28.6% of the controls. And this was a highly statistically significant difference. When we look at the separate outcome, looking for the spike protein, which sits on the outside of the virus, on the coat, we can see that the same trend is there, with 63.6% of the patients who went on to develop long COVID having spike protein detectable in their serum, compared with just 28.6% of the controls who had SARS-CoV-2 but didn't go on to get long COVID. So here's what the data look like. You can see on the left there are the data for detection of the genetic material, the RNA of the virus, and on the right the detection of the spike protein. And each column, for each figure, in blue are the people who don't develop long COVID, and in orange are the people who do develop long COVID. And you can see that the frequency of detection of these targets is lower in those who don't versus do develop long COVID, but also the levels detected are lower where they are detected. So the conclusions of this relatively small study from a single centre are that long COVID does indeed associate with persistence of the SARS-CoV-2 coronavirus. Now, on its own, of course, a finding like this can't be said to be definitive. What we really need to do is look at multiple studies, and there's a very thorough and interesting article just published in the Lancet Respiratory Medicine this month, which summarises these studies. And you can see there are no fewer than 12 studies now in people with long COVID, and controls who had COVID-19 but didn't go on to develop long COVID have found evidence that the virus persists in all different types of tissue, not just in the bloodstream, and is more commonly detected in people who go on to develop long COVID than those who don't. And the authors of this article, which incidentally is freely available online, go on to discuss some of the mechanisms by which persistent virus might be responsible for causing the symptoms of long COVID, be it in the brain, in the lung, in the cardiovascular system, so that's in the blood vessels, and in the gastrointestinal tract, so that's in the intestines, where persistent virus can cause prolonged abdominal symptoms, as we know are experienced by a subgroup of people with long COVID. So why is this important? Well, from a scientific perspective, of course, we need to understand what's going on, what's causing the presentation and the persistence of symptoms of COVID long after the acute infection. But from a very practical point of view, this finding that persistence of virus associates with an increased risk of long COVID opens the door to the idea that treatment with antiviral therapies could end up helping people to recover from long COVID. And I just highlight here 3 clinical trials, all being done in the US, one at Stanford, one at Duke, one at Yale, all investigating this combination of antiviral drugs, nirmatrelvir and ritonavir, in quite large samples of patients. And we look forward to the results of those trials as soon as they have finished. So that brings me to the end of the webinar today. I hope you found this of interest. Next month, I hope to have more data from our own cohort to share with you. But until then, thanks to all of you for continuing to post your data, complete the monthly questionnaires, and we continue to analyse those data to get insights not just into risk factors for acute COVID, but particularly now focusing on long COVID and trying to identify risk factors and potential areas where we could develop treatments for that condition. Thanks to all of you, and goodbye until next month.